All right, everybody, why don't we slowly get started? Hi, Bob. Nice to see you. So there's still some really good seats right up front. Excellent view of the screen. If you look kind of like you don't know what I'm talking about, I can see immediately, and I will be able to adjust what I say. Okay, so it's great to have you all here. So to our 2015 cell modeling workshop. And so what I would like to do is I'll just give a really brief introduction into why we're doing this and who we are. And then we'll briefly go, maybe we'll do some introductions. And then briefly go over the agenda to just kind of tell you what we have planned. And then we'll dive into MCell today, MCell and Cell Blender, and take it from there. So let's see if I can. Is this visible enough? Yes? All right. So as just before I get started, overall, please feel free to ask questions anytime. So we're trying to keep this pretty informal. So if there's something that's unclear or you have questions, so please feel free to interrupt. If there's too much interruption, I'll start complaining and just soldier on. But please, please ask questions. This workshop today is organized by an entity called the National Center for Multiscale Modeling of Biological Systems, MMBIOS. That's now in its third year. It's an NIH-funded P41 resource. And the goal of this MMBIOS Center that involves, let me see if I can actually advance this. It basically involves a whole bunch of different institutions. It involves the University of Pittsburgh, Carnegie Mellon, Pittsburgh Supercomputing Center, and the Salk Institute. So it's a whole bunch of people that are working together, and the main goal really is to develop methodologies that span multiple scales, basically going from the molecular scale all the way up to the cell level. And so actually as part of this workshop you see some part of this tool chain. Not everything, but some part. So the goal is really for us to look at ways to integrate methods and develop methods that go from the molecular level where people study protein dynamics or protein folding or protein function and structure or other biomolecules, all the way up to cell level systems like this part of hippocampus in the brain, all the way up to whole cells, and perhaps even further all the way up to whole tissue. So this is a connectomics type study where people are interested in the wiring diagram of how neurons connect in the brain, so basically based on EM reconstruction. And so the goal of today's work, so this is basically the scope of this center. So it's a very ambitious scope, of course, very difficult problems that we as computational scientists need to solve, and really how to integrate these methods. So that's not at all clear how that should be done or what the best way to do that is. But I think it's important because many of the approaches that we have, and you'll probably see this a little bit in today's or in this workshop, is that many of these approaches have sort of a target focus in terms of length and time scales, and it's difficult to really look outside this focus, and that this makes it difficult to really study problems comprehensively. So you're either tied to the cell level, you have to give up something on the molecular level, or you study the molecular level, then you can't go to large systems and long time scales. So there's always some sort of compromise that you have to make. So really how to make this into something that you can really study from the small to the large is a big challenge in the field, and I think something we need to address, and we hope we can address at least to some level. And so in terms of the workshop, so this is kind of a slide that shows a little bit of the workflow that we have tried to develop tools for to really help to go from the molecular to the cell level. And we'll touch on many of these things here during the workshop. So on day 1, which is today, we'll mostly focus on MCell and Cell Blender, which is looking at cell-level systems. But then we'll also address tomorrow mostly how to deal with systems that have a very complex biochemical reality, where it's really difficult to actually write down all the reactions because there are so many. So you have a combinatorial explosion of states, phosphorylation states, whatnot, and you can actually, it's difficult to even enumerate all the things that happen. So it may actually be impossible because it's too many. And then there's ways to deal with that using rule-based modeling approaches, and we'll tell you about those tomorrow. And then on day 3, we'll touch a little bit on a tool called Cell Organizer that really allows you to go from imaging data, fluorescence imaging data, for example, to develop actually models of your biological system, your cell, using machine learning techniques to, for example, train models that model the shape of your cells or of compartments, cell nuclei, endosomes, what have you. So their locations within the cell and also the locations of proteins. And you can actually use this pipeline that we've developed to feed that type of information into an MCell model. So you can use your fluorescence image data to train models and actually to use that to generate instances of MCell models to simulate these systems. So this is probably a little bit abstract at this point, but hopefully as we go along with the lectures, this will start to make more sense. Okay, and so this is the list of people that's involved. I don't want to go through the whole list. So you see a lot of people are involved as part of this effort, developing tools and methods. Most of them, I think all of them probably, will actually meet during the workshop today and over the next 2 days. And perhaps we should continue now. So maybe I will briefly go to the itinerary just to let you know. So basically today we'll start the morning off with mostly MCell-related topics, a basic introduction to what MCell is and a little bit to the language and the features. Then we'll talk a little bit also about the sort of theoretical underpinnings of MCell, what kind of the methodology is that MCell uses, and kind of what the theoretical basis for the algorithms that we use in MCell. Then we'll give you a quick or also a brief demo into Cell Blender, which is our graphical user interface to generate and create MCell models. Then hopefully we'll be ready for lunch. And then the afternoon will be hands-on tutorials next door. So behind this door is a computer lab where you can all sit down either on a workstation, or you're welcome to use your own laptop if you want, so whatever works best for you. And then we'll start with a bunch of tutorials, starting basic and becoming a little bit more advanced as the day goes on. And in the evening, so this is an optional session. It's mostly for people that are maybe interested in digging themselves into the MCell code base, perhaps developing some features themselves. I want to know a little bit more. So I'll try to give a high-level overview over MCell. And I'd also like to talk to people that have or need features that are not currently in MCell, and we can talk about these a little bit. So either we can guide you perhaps in how you can implement them yourself, which is not necessarily easy, or we're very interested in knowing what you need. So if there's anything that you need that we don't have right now, there will be a good opportunity for us to discuss this a little bit more, and perhaps would be something we can implement in the future. So that's, but that's optional. So only for those of you who really maybe are interested in that aspect. And so I won't go in much detail over day 2. We'll talk more about this tomorrow, but basically we'll focus on rule-based modeling a little bit, and then show you how to use these types of approaches also with software that we have developed, graphical user interfaces. One is called Rule Bender, and how that actually can be integrated into an MCell simulation. And day 3, again, as I mentioned, will be focused on these, a little bit on these generative models that you can actually use to learn or to develop models based on imaging data. In these afternoon sessions, you're also very welcome to work on your own projects. If somebody has a project in mind that they already started or are about to start, so that's would be a good opportunity for you guys to, if you have any questions or you started already and something isn't totally clear, for you to work with us and ask questions. So you're more than welcome to do that. So that's a good opportunity to get started with our help, and we can get a discussion going. And perhaps as even a collaboration, we can, if it makes sense. Are there any questions at this point? Otherwise, I would suggest maybe we'll go around real quick and introduce each other. It's always good for me and everybody else here to know who you are, and perhaps you can say in 1 or 2 sentences what your interest is. Maybe you have a research problem, or you want to use it for teaching, or just kind of what your interest in this workshop is and who you are. So maybe I'll start with myself. I haven't actually introduced myself. So my name is Marcus, and I'm sort of part of this MCell development team. I work here at the Pittsburgh Supercomputing Center, and I direct a group here called the Biomedical Applications Group that really focuses on developing biomedical software tools and then also applying these tools to biological problems. And so my background originally is in physics, and I've did a lot of MD simulations in my past life, so studying biomolecular structure and function, enzyme kinetics, things like that. And I've kind of moved over the last many years now to these more higher level cell level systems, and I'm very interested in those. Okay, so why don't we continue with you?
My name is Madison Brandon, and I'm
Okay, great. So that's a real diverse group. So we have people who are fairly experienced with MCell, and we have people that are very new to MCell. So I hope we can strike a balance. And again, if there's any questions, please feel free to interrupt. So the first talk, I think, is very basic. So hopefully there will still be something in there even for the people who have used MCell before. And the goal really is to just tell you what MCell is, and sort of what you can do with it, and kind of the basic structure of how to set up an MCell model by hand, actually, because it's kind of important that you kind of learn and understand a little bit, at least, the kind of language and the kind of infrastructure that's set up underneath to really run an MCell model. If you use that Cell Blender graphical user interface this afternoon, most of that will be sort of hidden behind the scenes. But probably once you get to more sophisticated models, perhaps at some point you will have to dive into the nitty-gritty details. It's kind of good to know what's going on behind the scenes. The slides will be available online, so there's no need for you to take notes if you don't want to. So we'll make those available. And we're actually filming these lectures too, so they should be on YouTube at some point if you want to watch them again, assuming the technology doesn't works as expected. Okay, so in the next hour, basically, it's basic introduction to running simulations using MCell. And I'll just start off with just providing a little bit of context. So what does MCell actually do? What's an MCell simulation, and what does MCell as a tool do? Basically, what MCell is, it's a Monte Carlo reaction diffusion simulator that's targeted at modeling systems that have some sort of complex or where the spatial aspect of your system is important in some aspect. And what that really means, of course, depends on your particular system, but it's specifically targeted at systems that are sort of at the cell level, micron to millimeter length scales. And you can use more or less arbitrarily complicated geometries. And so just to show you one example, and I think Tom will tell you more about this specific system in the demo right before lunch. So this is actually an MCell model geometry, or an MCell model that the Salk Institute folks have used to run simulations inside. So that's a piece of dendrite in the hippocampus, and you can see it's spatially very complex. It's from an EM reconstruction, and you can see very complex structural features. Here the synapses, there's lots of other structures in there, glia, what have you. And so for certain questions that you may have, the detailed structure of your system can be quite critical. And so MCell is, and if that's the case, MCell is really the tool for you. So it's really good at modeling these types of systems, yes.
So for me, not being used to looking at some of these things, having a scale bar somewhere in my head.
Right.
Is helpful.
So this is probably, Tom, half a micron?
The diameter of the yellow dendritic shaft is about 2 microns.
2 microns, right.
And the width of the hole, the width across the bottom is about 6 microns.
6 microns, right.
About 5 microns long.
Right. And so,
that whole thing would fit inside a single red blood cell.
right.
Just to give you an idea of how dense things are in the brain.
And so the main components of any MCell model, there's basically, you can classify them into maybe three categories. One of them is the geometry, and I'll talk briefly about how you would actually go about obtaining a geometry for your system. The second one is you have, in an MCell simulation, random walk diffusion of molecules within these spaces. And these can either be volume molecules, so molecules that are confined within these spaces, or molecules on the surfaces. They can all diffuse and react with each other. So you have molecules that are confined within these spaces that diffuse and react, and molecules on the surfaces that can diffuse and react. And you can have reactions between the two species. And then the third component is that you can have what we call biochemical state transitions. For example, this is a transition for an acetylcholine receptor, for example, on the postsynaptic side of a synapse that models basically the binding of acetylcholine to the receptor, of opening of the channel, and then conduction of calcium through the channel. And so these can be, again, very complicated and are modeled based on some biological insight that you have based in your system. So you can model any kind of open-close transition, or receptor binding, or conformational change that you have in mind. So these are the three key ingredients of an MCell simulation. And so the general pipeline, how you would go about developing an MCell model, is you have to somehow obtain or create your geometry. And I'll mention a few ways how you could do that in reality. Then I call this annotate meshes. And what this really means is you have to kind of tell MCell what certain regions on these meshes are. You could say, well, this is my synapse, or this is a region where molecules are released, or this is a region where receptors are located. So you basically kind of have to have a notion where things are within your model geometry. So you don't just need the geometry itself. You have to have some kind of notions where things are located within or on these geometries. Then you have to specify what I call non-spatial model parameters. These are things like the diffusion coefficients of molecules, the kinetic rate constants. And this is typically, for cell simulations, this is typically not an easy task, because often these numbers aren't well known. So very often, we just don't know what the on rates are or the off rates for many of these molecules. You know the KD values often. These are easy to measure, but the actual kinetic rate constants are very difficult to measure and are often sometimes tricky to obtain. Then once you have that, you basically simulate your model, and then you can visualize or analyze the results. And I'll talk about each of these points in more detail now. But this is basically the main strategy that you would go through when designing an MCell model. And quite often, it happens that there's some iterative procedure in this kind of flow, so it's often not linear, because sometimes you may not know certain parameters well, or you may have some quantities that you actually want to use these simulations to determine. So you would perhaps set up an initial model, you would analyze the results, you would compare with the experiment and see, oh, that matches, doesn't match, and then adjust some parameters, go back, simulate again until you have converged on some quantity for your parameter. Or perhaps it's a spatial component that you're after. You don't quite know, what the distance should be, so maybe you set up an initial model, you run it, you compare with experiment, it doesn't match, you move some things around, you move channels around, you move receptors around, you change the distance of something until you have some agreement with experimental results. And so typically, if you want to design an MCell model, it's useful and most of the time actually necessary that you have some kind of experimental data that helps you to develop the model. It's difficult to develop a model out of nothing, so that's quite tricky. But it's a very powerful tool in combination with some experimental data that you have to really help you test hypotheses that you may have, because it's much easier in a model to change numbers of things or locations of things, much easier than it is in experiment often. Right, so let's talk about geometries briefly. So how could you actually obtain a geometry that you can use for an MCell simulation? The first one is one that's been used quite frequently in the past, is to use actually electron microscopy data to segment and reconstruct a realistic geometry from your biological systems. For example, this is a neuromuscular junction of a rat, I think. So basically, you slice your sample, you take electron micrographs, and you reassemble that stack into a three-dimensional volume stack that you can then segment and more or less extract the meshes and import them into MCell simulation. So that's, for example, the image I showed of this hippocampal dendrite that was actually obtained with this kind of procedure. So somebody actually, and I see people smiling. And so this is a very powerful way because you have a really realistic geometry of your system. The main downside, of course, is that it's a very complicated and time-consuming thing to do. So you're talking about months, maybe a year, to actually get a single reconstructed system, depending on the complexity of your system. So it's not at all trivial to do. And so this makes this approach fairly time-consuming and difficult. But for those of you who may have such an image stack already or a reconstructed volume, this is a really good way to go. Maybe one other disadvantage of this approach, I'm not sure if it's a disadvantage, is that once you have such a reconstructed volume, you have one instance of how your system could look like, and it's not necessarily easy to change it. So if you wanted to investigate how certain geometrical parameters affect your system, it's not so easy necessarily with a model of this kind, because it's just too complex to really say, well, what happens if the extracellular volume fraction changes by 10%, say. It's not so easy to do with a model like that. So I guess maybe what I'm trying to say is that depending on the questions that you have, this approach may or may not be good for you. And that's, I think, true for any of these approaches that you could take, that it's good to think about what's the actual biological question or insight you'd like to gain from a simulation like that. And that will dictate, at least to a significant degree, which method really is good for you to use. Then the second approach, and that's one actually you will learn about during this workshop, so I don't want to talk about it in great detail, is to use fluorescence microscopy imaging and the cell organizer tool to learn generative models of your system, and to use that actually to feed MCell simulations to create instances of your model with locations of whatever proteins or intracellular compartments you're interested in. And so we'll learn about this during the workshop. So this is a neat way for you to develop cell simulations if you have sort of fluorescence image data of your system. So this actually, I think, is quite powerful. And then the third approach, which is maybe is probably the simplest one, but can also be quite powerful, is that you actually use you construct a geometry in silico using CAD tools, for example. And this is what you can actually do with the Cell Blender tool that we've developed, which you will do this afternoon. So you basically develop your geometry from scratch based on data you know from experiments. So it's by no means a hand-waving type of approach. So you base your geometry on real measurements from experiments. For example, in this case, this is a model of a neuromuscular junction that's based on freeze fracture data from freeze fracture electron microscopy and other experiments. You can really, so you can, in this case, measure the distances between these rows of synaptic vesicles, and you can make actually a fairly detailed model using these approaches in the matter of an hour or two. So it's actually a very fast and effective way to develop quite complex and rich geometries. And one big advantage of this approach actually is that it's fairly easy to change geometrical parameters. So once you have a model like this in Cell Blender, it's very easy to say, well, what happens if I change the number of vesicles? I just remove some. Or what changes if I increase the distance between the voltage-gated calcium channels by 10 angstroms? You'll just move them, save the model, and run it again. So it's very easy to explore geometrical parameters using an approach like that. So if you're interested in these kinds of questions that you're sort of trying to probe, how does geometry affect my system, and you're not really necessarily, at least at that stage of your investigation, interested in the intricate 3D structure of your system, then that's a really good way to go. So it's quite, and you learn how to do this afternoon. It's very quick, so it's really, really easy to do. And any questions about sort of how you would go about creating geometry? There's certainly more, so it's not, so you could envision other ways to get geometry into MCell. So MCell basically just expects a description of your mesh, which is just vertices in space that are connected. So I know that people have written MATLAB scripts to create meshes, and you can provide a Python script, or there's certainly many other ways to get meshes, but these are sort of the typical ones, but this is by no means comprehensive. Any questions about geometries?
So as the annotation of that, because you haven't actually gotten into annotating these bases yet.
Right. So basically, if you would have a realistic, if this would be a geometry, then you'd had to, after segmentation, you had to kind of define, maybe where the postsynaptic receptor is located. So that's not clear just based on the geometry. Just tells you how it looks like. It doesn't tell you where stuff is. So the next and perhaps complex step would be to decide, where should I put the postsynaptic receptors? Where should the vesicles be located? Where are the calcium channels? Where was the other thing. So that's basically the annotation. And that doesn't typically come out of the imaging per se in this case. So that's something you have to know by other means. In this case, so this approach helps you here because cell organizable, this pipeline will actually tell you, where are my proteins located, perhaps as a function of time in space. So they're here, here, and here, or in vesicles. So this pipeline may, if you have these fluorescence imaging data, you may have an easier time here because you actually you have that information. You know where stuff is in the cell based on fluorescence tagging. And I guess, and here again, you have to, in this case, I think it's fairly clear. I mean, you have to know where stuff is in the first place because you're building it from scratch. So in a model like this, you presumably know where are your important ingredients. But it's something one has to think about. As soon as you go spatial, you have to know a lot more about your system. You really have to know how it looks like and where stuff is. For people that are used to ordinary differential equations, you can just plug in a concentration of your, certain concentration of your stuff. That's not good enough here anymore. So you typically have to know where things are in space because that's often critical, actually, for the biology of the system.
I'll say a few things about it.
Sure.
During my talk.
Oh yeah. Okay. Great. Yes. And perhaps if you guys have models in mind that you would develop, and you're not so clear exactly what that means for you, please talk to us, and we'll try to help you or see if we can figure out how this could best be done. And sometimes, actually, going back to this pipeline that I talked about, sometimes the location of things may be the question that you have. Maybe the question is, where you have some experimental constraints, you know the basic geometry, but you don't know where your receptors are located. And so maybe you can use the model to play with that. You say, well, what happens if I place them here? Does that reproduce the experimental data that I have? No, it doesn't. So maybe that's not where they are. So maybe they're confined somewhere, or maybe they're localized, or maybe they diffuse slower in this region. So this may in fact be a question that you have for the model. Where's my receptor located, and how does it behave, so that I can actually reproduce the experimental data that I have. So it's quite possible. And so that goes back to that pipeline that I talked about before. Often you have some kind of question, you have some kind of unknown in your model, how many of something, where something is, the exact way something works. And often you do this iterative procedure that you put in a first guess of your best guess that you have. You simulate it, you analyze, you compare, and then you realize it doesn't work, or it works, and then you change. And then you maybe converge at some point, hopefully.
And as Marcus mentioned a moment ago, maybe that question is your question, right? Maybe that is the unknown thing that you're trying to discover through your model. And we've actually had some success in this approach. Instead of lamenting your predicament of not knowing where things are, embrace it. And we've published a couple papers where we've made a prediction from our modeling that the only way you could get the experimental result that had been measured was if the distribution had been was a certain way. And in one case, it was completely contrary to the dogma, but no one had experimental evidence to support their dogma. And our simulations make a startling prediction, and another researcher made it his mission to prove us wrong, and ended up proving us right and becoming a close colleague.
Right. So yeah, so that's a very good point. So this is really So I put the geometry first. So that's typically how when I think about new systems, approach new models. I think about the geometry and where stuff is first. But that's not necessarily the case for everybody. But it's good to think about, if you design a spatial model, it's good to think about the spatial aspects of your system first, and start designing it from that perspective. Yes? Yeah?
Just a short question.
Mhm.
I think I will talk to you later for that. But is there a way to, let's say you have an image stack,
Mhm.
a 3D image stack of a cell, and usually you could just use ImageJ to create a 3D model, and you can import it in Blender and use it in Cell Blender, and you're totally fine. But is there a way to avoid Blender and Cell Blender? Just you have your diagram of the stack in a 3D model, and you analyze this through whatever, I don't know, and then you have your geometry MDL file.
You mean you're basically asking if it would be possible in principle to output the MDL directly from ImageJ? Or
Yeah.
it would definitely be possible. I mean, in fact, we can talk. So I know that you can write plugins for ImageJ. I'm not familiar with it very much. But I mean, the geometry format that MCell uses is very straightforward. So if you have it within ImageJ, you could provide the script in a few hours.
The only two MDL files I cannot write down on my mind is the initialization and the geometry files. Those two are
Yep. Let's talk. I think this should be fairly straightforward to do. It's a simple mesh, so I don't see any problem with that. Okay, so now hopefully in your task of developing an MCell model, we have some kind of geometry. We know where stuff is, and then we'll need to define a few more things. All of them probably fairly obvious. First of all, we'll have to define some molecules and give them some properties, and the key property is the diffusion coefficient in this case. So we'll define molecules. We'll define where they are. They could be in the space defined by your geometry or on the surface. And more specifically, they could be in specific regions within your space and in specific regions on your surface. And you can do all of that. So you can place them wherever you want, in whatever region you think they are. And so you need to know their diffusion coefficients, and you need to know the concentrations or densities of your molecule. So that's something you'll have to know in order to actually put them into your simulation. The next thing that you need to define are, typically there's reactions in your system, things interacting, not necessarily. So for people that are just interested in diffusion, they may not actually have reactions, but typically there's some kind of reactivity in your system, either between these volume molecules or between surface molecules, or you have these state transitions that correspond to opening and closing of channels or binding and unbinding of receptors. So you would need to define those. And as I mentioned already briefly before, sometimes this can actually be a difficult step in the sense that depending on the kind of problem that you have, it can be quite tricky to write down all these equations. If you have a molecule that has many phosphorylation states, it can all be phosphorylated or unphosphorylated, to actually write this down as a series of elementary reactions that can quickly become 2 to the power of, and you can do the math, reactions. So this can become quite complex and complicated. And this is where BioNetGen really can help you a lot. So this is a different way of specifying reactions by defining rules that describe what can happen in the system. And Jim will talk a lot about this tomorrow, so I won't belabor this right now. But if you have a system that is very complex and it will be very difficult to actually write down all the equations by hand, then BioNetGen could really be, or is most likely the tool you want to use to deal with that complexity. And then the third key thing that you want to specify is you want to specify some output. And again, output is a little bit more complex in a 3D reaction than in a non-spatial reaction, because you can count molecules or visualize molecules not only based on their presence, but also where they are in space. So you might want to count how many molecules are over here versus over here, how many molecules are close to your channel, a little bit further away from your channel, a little bit further away from your channel. So maybe you want to have the concentration profile as a function of distance from your channel. And MCell basically gives you all the flexibility that you could ask for, I think, to really measure whatever you want. And so you can set up very complex measurement scenarios to really query your system. Where are my molecules? How many are there? And where are they located?
And you can even ask when and how often
Exactly, yes.
things can happen. When, where, and how often.
Right. So after you have done this, so you have defined your molecules, you have told MCell where they are, you have defined your reactions, you have defined your output. Then the next step is basically just simulate MCell, your model. And one way to do this is to use Cell Blender, and you'll do this afternoon. So you can actually simulate your model in Cell Blender by just clicking a button. But if you may be like me, who still likes the command line, you can also run it from the command line. And so the way you run it is basically your model is defined in this file. It has this extension MDL, which stands for Model Description Language. And I'll talk more about what's in this file. But to run it, you basically just say MCell and then mymodel.mdl, and MCell will run it for you and produce whatever output you've requested. OK. Right, and then you can visualize your output. And our visualization tool of choice is Cell Blender. So you can import your output. You can see where all the molecules are. You can make movies, screenshots, whatever you want for publication. So it's a very convenient tool for all of that. It's very simple to do. And Bob and Tom are the experts. And Jacob. I'm not. Typically, at least for the kinds of things I do, the key output is really reaction data output, where we count numbers of molecules, numbers of reactions. And MCell will right now just give those to you as plain text files that you can analyze in your favorite tool. So you can use R, Python, Octave, MATLAB, whatever you have. So it's depending on the kind of on the amount of output that you have, it's probably good to develop some kind of automated way to analyze your data in a simple fashion. So I use R a lot personally. But people like to use whatever they like. Yes.
Would you please explain a bit about the simulation engine, the mechanism? All I know is that it's a Monte Carlo.
I'll talk a little bit more about this in the second half. So give a little bit more of the so what's really going on. But if it may not be detailed enough for you, then we can talk. I'm more than happy to talk to you about it in more detail.
And I think this evening.
Sure, this evening, right. Right, right.
Then it will be at the level of.
Right. OK, so are there any questions still about this? So this was just the basic simulation workflow geometry, anything that's non-spatial, molecules, reaction, output. Then you simulate, then you analyze, and then typically you take some action based on that. You're happy, or you say, well, I'm not happy, I need to go back. Yes?
I would like to know how many, because what I understand is this software converts static images into a simple model or a movie. So what is the limits or what's the requirements of these input images, size of the input images, and then what is the final size of the MDL files typically?
It's really difficult to give a good answer because it really depends on the model. So I mean,
How long?
so you know, if you have a complicated geometry, then you will have the MDL file that actually represents your geometry will be fairly sizable, right? Because you have many vertices that need to be described, and then the rest depends. You know, if your model has 5 reactions, then your reaction data file will be, you know, 5 lines long plus 2 and 2 at the top and 2 at the bottom. For example, the models that I run, you know, looking at the neuromuscular junction, there we keep very detailed track of, so we have voltage-gated calcium channels in this simulation, we have vesicles that bind calcium, and we actually keep very detailed track of which calcium ion from what channel binds to which vesicle. And actually even a little bit finer grained than that. And so we have very detailed bookkeeping requirements. And so, for example, our output specification file tends to be sometimes 250,000 to 500,000 lines long. Right? So they're very extensive. I mean, they're all automatically generated, so we have scripts for that. But they can be very long. So it really depends on what kind of questions you ask. If you have, you know, many molecules and you really want to know, and you have many channels and you really want to know where did this ion come from that just bound here. I really want to know which of these channels it went through. Then, you know, this can be complex. So I guess my answer is there's no good answer. So I think typical MCell models are fairly compact, but they could be large, and it really depends.
And the model that I'm going to show you consists of about 4.5 million polygons for the mesh, and runs with good efficient partitioning of the thing, runs at about 4 gigabytes of RAM. And for the simulations that'll show you, a single run runs in several hours. But because it's stochastic and you know and highly variable, the type of things that you know that we studied for synaptic release, vesicular release, is probabilistic and has a low probability of release. It's highly stochastic. You need to run many. In fact, that stochasticity is what you're studying. Right? So embrace it. Right? Like I said, you typically need to run hundreds or thousands or tens of thousands of simulations of a single instance of a model with different seeds in order to characterize what is the precise probability of release, and is there we study facilitation and depression of the system. And so to characterize that, you might have to do 10,000 simulations of the thing. And so a typical output from all of that might be close to a terabyte of data. And then after that is your analysis of that data, which is a whole other thing using what your favorite analysis technique, right? And you can analyze that in parallel, often on a parallel supercomputer to pipeline all that.
So we can talk also a little bit about this in the afternoon. I think sort of a simple model in MCell is actually quite compact and very fast, so it'll probably run in a second or 10 seconds. As Tom mentioned, if you...
This model that we're talking about, the MDL for the whole thing is like 200 megabytes. The geometry is like 180 of that.
Yeah, so I think typically the models you develop in CellBlender will be fairly simple, but it can become quite sophisticated. And again, that depends on the question that you have.
Oh,
And depending on the...
starts up in about 30 seconds. Right, so very efficient to start it. After that, it just runs.
Yeah.
It also depends a lot on how long you want your diffusion time to
Right.
take. Sure. The longer it is, the further the molecules get.
Right.
The model has to get bigger, so our models are usually gigabyte of
Yeah.
MCL file and 3 weeks of run, just because they happen to be long diffusion times. So
Right.
right.
Yes, that it's for people that have big models, we can also talk a little bit and see if there's maybe something we can do to help. So actually for our modeling, we have a custom binary output format for the reaction data, simply to make that more compact. Because as I mentioned, we have a lot of output, and so we have basically tens of terabytes of output data, and it's sort of a compressed format that's very effective. So we could, it's not generally available, but we should talk. So for people who have big models, you have to think a little bit more about your pipeline and how do you analyze the data. It can be a lot of data. And so I think that's in general, as you develop your model, it's always good to think about, what is really the question that you have, what should you keep track of, and make sure that you measure all the necessary quantities, but ideally not that much more, because it can really add up if you need to run, if you do your statistical sampling or if you run for long times. And so this is, Tom mentioned this already, I just want to emphasize it again. So these are stochastic simulations, so you have to do stochastic averaging. So there's no point in doing a single MCell run typically, unless you have lots of molecules in there and the sampling is sort of done just by sheer number of stuff. But typically you want to do some averaging over some number of runs. And how many runs is not, I don't think, at least I'm not aware of any easy answer how many you need. It's typically based on the quantity that you're measuring, and you'll just have to run maybe 10 or 100 first and see if things converge, how things look like. If they look converged, then you're probably okay. But often you may have to run more, so that's good to keep in mind. And so I sometimes see people just doing one MCell run and reporting a number. That's typically not good, because you just don't know. I mean, you may have accidentally picked the 1 point way out in your distribution that's really not representative at all of what you're looking for. So typically you want to run some more, at least to get some confidence in that you have your measured quantity is really representative of the average behavior. And in fact, the fluctuations are quite important. So they actually provide a lot of insight in your system and what's really going on. Okay, so I'm of course way behind, so I will try to speed up. One thing though I wanted to mention, so for those of you who know the story, I mention this every year, is the satellite here, Mars Climate Orbiter. I'm not sure if people have heard of that, that crashed and burned now quite some time ago due to a very trivial reason, namely one part of the team was using SI units and some other part of the team was using imperial units, and they didn't know about this, and so they were of course off by a factor of 2.2, but that was good enough to basically destroy this thing. And so don't let your MCell model become the next Mars Climate Orbiter. So units are really key to know about and to keep track of. And it's unfortunately, especially if you dig through the literature and look for values of stuff based on somebody's experiment, it's good to be very careful with the numbers that you extract and make sure they have the right units. The units that MCell expects, and I'm just going to list them here. So spatial dimensions in microns, time is in seconds, diffusion coefficient centimeter squared per second, and so forth. So you can read it yourself. So be very careful about making sure that the numbers you use based on experiments or literature, that they have the right units, and if not, convert them. Unfortunately, there's a lot of incorrect results out in the literature. So if you extract values from papers, I would always try to confirm it with a second paper if you can. So there's a lot of, unfortunately, at least in the field I'm working in, there's a lot of bogus numbers, and units are wrong, and things are just all over the place. So if you have one key parameter that your study hinges on, just using one paper for that is tricky, because so I would so be careful when you actually take values from the literature. It's really, unfortunately, a mess. And it's a mess because there's no real agreement on what the right units are. So you'll see MCell has these units. You probably have other tools that have different units, and people. So it's a little bit of a jumble, and it's unfortunate. But the only thing I can recommend is to really be careful and just make sure that the numbers you use have the right units that MCell expects, and double check or triple check if you can. Okay, so what I wanted to do is maybe before we break for the next 20 minutes or so would be to briefly walk you through some basic MDL, where MDL is model description language that MCell uses that you use to describe an MCell model that MCell can actually then simulate. So it's going to be very quick and brief walkthrough. There's by no means an expectation here that you memorize everything or understand everything, just to show you the key concepts. As you go through the tutorials this afternoon and work through some examples, I think many of these things will become much more clear. But I just wanted to make sure that you kind of understand the basic concept behind this model description language, which is basically what tells MCell what to do. And even if you use Cell Blender under the hood, there will be these files generated that MCell then uses. And for those of you who have done some programming or write some MATLAB scripts, it's basically a programming language in some sense that describes the system to MCell. And it consists of certain keywords or commands that are always in all caps, so like some command equals some value, or consists of block statements of this form. So this is a typical, this is how MCell commands are specified. And so what I would like to do is walk through the creation of a basic model that doesn't do anything profound or significant, but it kind of shows some of the concepts. And again, if there's any questions, please interrupt at any time. So
I should also say it's not XML, right? It's meant to be human.
right. And it's quite human readable, actually. So we'll start off. So this model is called firstmodel.mdl. You can do this in any text editor that you have. Ideally not Microsoft Word, so just some text editor. And if you don't know which one to use, we can help you. There's a lot available. For the OS X users, I think there's Text Wrangler or something, which is pretty good. Or even TextEdit works. So this is the start of our file. There's two MCell keywords you can see here. They're all caps, iterations, and time step. And these you always need, basically. The number of iterations you want to run and the time step in, what's the unit for time? Seconds. Excellent. Right. And just to show the concept, you can define variables. For those of you who are sort of used to programming, so you can define some variables, like iters would be our iterations, dt would be our time step, and then you can use them in the MDL actually to define things. That's sometimes useful. For example, you might want to use the time step in multiple places in your file. Then it's good to define this at the top. So if you change your time step, it's changed everywhere and not just in the one place that you happen to see. It's a common mistake. So this is how we start our file. You can put comments in your MDL file. And the comments are like, for those of you who are C programmers, it's just like in the C programming language. This starts a comment, this ends a comment. And I would actually highly recommend that if you design a model, comment it heavily. I mean, really put down, why you did this, why this reaction is that. You might even want to put down where your k values came from or your diffusion coefficient came from. You may remember it now, you may remember it next week, but if you look at your file next year because somebody contacts you, oh, I saw your paper, why did you use this constant? Then you're basically staring at a file that says nothing, and you may not remember anymore. So it's a good thing if you design these models to put comments in that just tell you for the future where things come from. So I do that a lot, but you can also put it in your lab notebook, but it's easier to do in the MDL if you want to. So this is Cell Blender. You'll get to use this afternoon. And so I'm going to do something very simple here. I'm just going to use Cell Blender to create a cube. Not a very biological system, but good enough for us. And basically save it as an MDL file. So you can do that in Cell Blender. This afternoon, you'll actually design your whole model in Cell Blender, so you can do that too. But for now, we want to actually stick to making this model file by hand. So basically, I use Cell Blender just to create this cube and then export it as a file called cube.mdl that contains the description of how this cube looks like. And we'll look at this in a second. And so this is the next concept that's quite useful in MDL, is that you don't have to have one gigantic MDL file. You can actually split it into pieces, and you can include them into a main MDL file. That's typically what we do. So you would have an MDL file that contains all your geometry. You have an MDL file that contains all your reactions. You have an MDL file that contains all your molecule definitions, and so forth. That really helps to structure things. And often, the geometry files tend to be very large and complicated. It's not something you ever want to look at all the time in your main file. It's just something you have and typically never changes. So you just want it to be out of the way. There's one small comment I should make. So these includes are lexical. So basically, these things are included when the parser encounters them. So there's a little bit of an ordering dependence. Right? So you can't define your reactions before you haven't defined your molecules. So MCell has to know about the molecules first. Otherwise, it doesn't know how to do reactions with molecules that it doesn't know about. So some things are a little bit, so you have to just watch out for that. But it's usually fairly obvious how to do that. So this is actually how this cube.mdl file looks like. It describes the cube as a list of, so it's a polygon list, which is how most geometries are described in MCell. And the format is fairly simple. It's a list of vertices in x, y, z coordinates. And then a list of element connections that define triangles. So basically, it connects vertex 4 with vertex 0 with vertex 3. That defines a triangle. And so these element connections here define triangles that describe the cube. Right? Does it make sense? So basically, you have vertices. These are just x, y, z coordinates in microns. Right? So the units are microns. Space units are microns. So we have a cube. So we have 8 vertices, right? 4, 4. So we have 1, 2, 3, 4, 5, 6, 7, 8. And element connections, so we have how many triangles on the cube? So we have 6 faces, right? And each face is divided into triangles, so we have 12 triangles. So these are the 12 triangles that describe each a triangle of this form, right? So you have a vertex 0, 1, 2 that defines a triangle. And so one important thing to notice that these surfaces have an orientation. So you have, in this case, if the triangle is 0, 1, 2, that defines a right-handed orientation. And that actually defines the normal vector for this triangle. So if you take a right-handed rule, if you go, so if the order of vertices for this triangle is 0, 1, 2, the normal vector would point in this direction. Does it make sense? So that's actually quite important, that your mesh has a consistent orientation. So in particular, neighboring triangles have to have, of the same object, have to have the same orientation. So you can't have a triangle that has the normal vector down next to one that has it up. That wouldn't define a surface. This would actually be 2 separate objects in a way, right? There would be a gap in between. So if you use Cell Blender, you don't have to worry about it. Cell Blender makes sure that it's all uniform. But if you get your mesh from a reconstruction or segmentation, you will have to deal with that. Most likely, if you get that thing back from your segmentation software, it may be a mess. It may be, because from a visual point of view, it doesn't matter. It's just a surface. The normal vector, typically you can't see it. But if you wanted to convert it into an MCell mesh, you have to make sure you have consistent triangulation. So that's quite key. So again, if you use Cell Blender, you shouldn't have to worry about this ever. But if you get your mesh from somewhere else, or you write your own script, I've actually frequently people, submit bug reports. They have a MATLAB script that generates the huge geometry, and then they start it up, and MCell complains that something's not right with their surface. And it's typically that some triangles that they generated have the wrong order of vertices, which means that the normal vector points in the wrong direction, and then it's just not right.
There's a couple widgets inside of Blender.
Mm-hmm. Right.
Yeah,
Good point.
Find and pressure like that. Typically, you want to make sure that your software in your pipeline of generating these things is doing the right thing.
Right. Yeah, so in Cell Blender, you can check if your mesh is what's called watertight, which basically means it has a consistent orientation. Right, so basically we have a vertex list and list of connections. That's all that describes a surface. And if you have a more complex surface, it's just more vertices and more connections. All the surfaces are subdivided into triangles of that sort. So this is really all there is in a mesh description in this MDL file. And it's basically you could use a script in ImageJ to just output that if you have your vertices. Again, making sure that your triangle orientations actually have the right normal vector. So we can talk more about this after. And so in addition to just defining the cube, so we have our 8 cube vertices and our 12 cube triangles describing the sides of the cube. We can also define regions on this object, and regions are basically just collections of triangles. So you can make any kind of region you want. So in this case, I have a region called top, which it happens to be the 2 triangles on the top surface of your cube. But you could pick any collection of triangles to make any kind of region that you want. If you have a more complicated mesh, you might pick the triangle that corresponds to the location of your presynaptic receptors, or it could be a round region on your cell that corresponds to a lipid raft. So whatever, and you can use Cell Blender to very easily define these regions. You can basically, once you have your geometry defined, you can paint on the surface to say, okay, this region is my receptor release site region, and this region is my channel location region, and this region. So it's very easy to define whatever spatial structure you want to have for your geometry. And so the beauty of Cell Blender basically is that this all happens automatically. So you see it in front of you, you define your regions, you press export, and then it's done for you. So you don't have to do this by hand. It would be quite tedious to do by hand, albeit possible. So. But just so that you kind of see how this looks like. But this orientation is really a key thing. So orientation is really a key feature of surfaces in MCell, and you can actually use that to transport stuff across a surface either way by means of that orientation of the vertices. And I'll talk about this briefly. Any questions about this? Again, this is a lot of detail. Often, if you use Cell Blender, you may not ever need to look at these files, but it's good to know what's in them. Okay, right. So, so we have basically now this cube file that we just saw that defines a cube object, and then the next key step is to define an object that's instantiated. This is the actual object that will be part of your simulations. You can view these geometries that you have sort of as templates. You can define as many templates as you want, and then add them to your simulation as you wish. So for example, you could define this cube object. And what we do here is we basically just add it to our simulation. We don't do anything to it. But what you could do is now you could actually add this cube object a couple times, translating it, rotating it if you want. So you could define one cube, then you add it, then you translate it. You add another object that's translated by 5 microns in the x direction, in the minus x direction. So you could actually use this one cube object to define an actual simulation that has hundreds of these cube objects that are all translated. And
So
yes.
the cube on the left, it lives in a different namespace than the cube
Yes,
on the right.
yes, right. So this is basically just a template geometry, and you can translate or rotate it as you wish. So you could build up actually a big system that has hundreds of cubes in them.
Probably a good idea to name it differently, maybe.
Yeah, so yeah, if you would translate them, you would name it cube 1, cube 2. So this basically imports this cube object and names it cube again, which if you just add it once, it's okay. But if you want to add 100, you would name this one cube 1 and then hundreds more. In
The instantiated one here, since the parent object is world there.
Yeah.
The instantiated object would be world.cube.
yeah.
Right.
So this is I think how we used to do how we used to build up a lot of systems. Now that we have Cell Blender, a different way is just to use Cell Blender to create separate cubes. You could do that too instead of using that feature. So both ways work, and probably most people would just use Cell Blender to create 100 different cubes instead of creating one object and then shifting it around. But both ways are possible, so there's no okay, so basically that defines our geometry. So the next thing we need to define molecules. So we have our geometry. We want to simulate in our cube. And so next we add some molecules. And adding molecules is very simple. There's a block called define molecules. And then you just list their names, and then in brackets their diffusion constant. And it can either be a 3D diffusion constant that makes this a volume molecule, a molecule that diffuses in the space, or you can give it a 2D diffusion coefficient that defines a surface molecule that lives on a surface. So that's basically how you define a volume versus a 2D molecule. So it's very simple. And the names are basically free form, so you can choose whatever makes sense for your simulation. I would recommend to avoid all caps names, because all caps stand for a typically MCell keyword, so it's confusing to do that. If you don't actually use an MCell keyword, it's probably fine, but it's just better to not use all caps for names of molecules or reactions. Does it make sense? So very simple. Just define them like that. Again, diffusion coefficients are centimeters squared per second. There's other units, micron squared per millisecond, and so forth. So make sure when you get the diffusion coefficients from literature that you do the right conversion, which in this case is simple. All right, so basically what we've done now is we have defined molecules. Now we have to actually tell MCell what we want to do with these molecules. And one thing we might want to do is actually to release some in our model. So to release molecules, we have to tell MCell two things: where to release them and how many of them. So these are the two key things. It's the minimum you need to tell MCell. And one way to do this would be actually to define what we call release sites. And release sites release molecules typically at the beginning of a simulation, but there's actually ways to release them at later times using different temporal patterns, so you can be fairly complex with that. But most of the time, we release them at the beginning, but it's not a requirement. So there can be more complex release patterns if you need to that can even be triggered by reaction. So it can be very complex, and we can talk more about this if you're interested. Yes.
It's like a continuously releasing source easily.
Yes, right.
Does
Or-
in order for a surface molecule to be confined to a surface, is that instruction contained when you say it's a 2D diffusion protein? That's what
Right.
ensures that it stays on the surface.
Right,
and
right.
doesn't go into diffusion.
Right, right. And so we have two release sites here. One releases this volume molecule called vol1 that we defined up here. In this case, we release a certain number. We could release by concentration too if you wanted to, but in this case, we release 2,000 of them. And we have to specify where, and we specify this by giving the release site a shape. And in this case, the shape is just the whole cube. So this is how you actually specify the cube. The world object is the main parent object, and then within the world object, the cube. So world cube is just our cube. So in this case, we tell MCell, please release 2,000 volume 1 molecules within the cube. And we also want to release some surface molecules. So we want to release the surface molecules, again, 2,000 of them in this case. And surface molecules we have to release on a region, not inside the object, but on the surface of the object. So we could say, again, in this case, world cube, and that would default to the whole cube. So it would be released on the whole surface of the cube. In this case, we actually specify this a little bit more succinctly. We actually say we don't want to release it on the whole cube, but on the top region of the whole cube. So if you remember here, we actually specified a surface region called top, which was the top surface of our cube. And so the name, I named it top, so the name is up to me. I could have named it anything. And so, yes. Maybe it was the bottom, who knows. So basically, we say we want to release these surface molecules on our cube on the region called top. And so you can see, even though this is a simple case, in this case, it's just two triangles, the top of our cube. It could be something very complicated. It could be any kind of region that you have defined on your very complicated geometry. So it could be, you know.
It could even be a Boolean
Right.
set operation on a combination of.
Right. So there's quite a lot of flexibility. As Tom mentioned, you can do Boolean operation. You can combine regions or intersections of regions. So it's quite flexible. So it's likely that you can specify any kind of region that you have in mind quite easily. Yes.
If you define multiple, let's say, triangles not connected to each
Sure.
other as one region, so
Sure.
you have multiple regions within one region variable?
Yeah.
Yeah.
Region is basically just,
Have to be contiguous.
Yeah.
It can be disjoint.
it's anything, whatever it is that you're after. So it's just a collection of triangles in a way. So it could model anything you want.
And then self-blender makes it very easy to.
Right. Right. Yeah, so these are things that are visually quite straightforward because you can see it, you can say, oh, this is the region. And so for that, cell blender is really key. So this is basically one good way to release molecules, to define release sites, either for volume molecules or for for surface molecules, and then release either by number or concentration / density, depending if it's a 2D or a 3D release. Yes?
Is this initialization, or is it number per sec?
So this will be, if you specify it like this, you will release 2,000 molecules at the beginning of the simulation. They will actually be placed randomly in the cube. So you'll have a random distribution of volume molecules in the cube, or a random distribution of surface molecules on this region on your surface. So it's not going to be patterned. It's going to be a random collection of molecules. But again, if you don't want to release them all in the beginning, there's ways to have sort of a temporal behavior to that, depending on the questions that you might have. Okay, so I wanted to just touch on one thing briefly, which is actually quite a little bit complicated, even for me sometimes, because I don't, but it's quite powerful, and so I wanted to mention it now, and we can talk more about it as we go along, and there's some tutorials actually on that as well. And this is relates to surface molecules, and actually what you can do with the capabilities that MCell has with orientation and sidedness of surfaces and surface molecules. And so for surface molecules, and I picked a random one here. Some people might be able to guess what that is. If not, I'll leave it as a... So that's KCSA, so it's a potassium channel. And so this kind of indicates so this is a membrane channel, right? So it conducts potassium ions through this pore here. And this sort of indicates the location of the membrane for this potassium channel, right? So the channel would actually sit in the membrane. The membrane would sort of end at these positions. And so in our MCell MCell has a notion of for surface molecules of the top and the bottom of them. So basically, surface molecules have a top and a bottom domain. And what that really means is not specified. It just means that you have something in the surface, and it has a top and a bottom. And what that specifically means depends on your molecule, right? So for the channel, the bottom could be the cytoplasmic side of your channel. The top could be the extracellular side of the channel. For a receptor, it could be different. Key is there's a distinction between these two sides. And what that means biologically depends on whatever this molecule represents in your simulation. In this case, it would present, the bottom would present one side of the channel. You can see it's not a symmetric protein, right? It has two distinct sides. And one of them would be bottom, and one of them would be top. Then the second concept is that, I mentioned that membranes have a normal vector associated with them. So they actually have a front and a back side. Right? So the front side is where the normal vector comes out. The back side is where the normal vector points into. So we call that front and back. Is that everybody clear with that? And so that means that there's a couple of ways to actually put molecules, surface molecules, in a surface, right? So this is our surface, front and back, and our molecule has a top and a bottom domain. So I can either insert my molecule with a top domain at the front of the surface, or with a top domain at the back of the surface. So I have these two possibilities. Right? And so that means if you actually place surface molecules on a surface, you have to think about how they should be placed on the surface depending on the kinds of question the biological question that you really have. Perhaps the orientation doesn't matter to you. Then you could just say, well, I don't care. So just put it randomly. You can tell MCell to put it in randomly, but typically you do care. And so you have to be aware of how you place molecules on the surface. One thing I should mention that's sort of important under some circumstances. So what MCell actually does is it subdivides it subdivides your surface into tiny triangles. Tiny meaning, so they're not the triangles that you see in Cell Blender. It actually subdivides them much finer. And the density of the subdivision is controlled by this keyword called surface grid density. By default, it's 10,000 per square microns. And so each of these small triangles basically has a surface area that typically corresponds to a surface area that you would expect from a typical membrane protein. So if you look at a membrane channel, or a membrane receptor, then they typically have that kind of surface area. And so that means that if you start putting molecules on surface in MCell, there's an upper limit of how many molecules you can actually pack on a square micron of your surface. Namely, by default, you can pack 10,000 molecules on the surface per square micron, right? And so what that means is that there's some kind of crowding taking place on surface in MCell. So if you have a lot of molecules on your surface, they're basically packed so densely that in a real biological context, it would basically be just protein. There would be no space anymore for lipid, so something like that perhaps. Then your simulation will start to become crowded. At some point, MCell will tell you, I can't place any more molecules. You basically filled up the mesh. So one remedy for that would be to increase the surface grid density. But if you do that, you have to kind of think about what this really means for your system, because you basically are reducing the surface area that you allow each of your molecules to have beyond what you would typically expect a membrane protein to have. So that's not necessarily wrong. You just have to think about, if you really need to pack it more tightly, what that really means for your system specifically. So most of the time, that's not an issue, but some people run into that when they really pack a lot of molecules on the surface. But again, the biological question arises, what does it really mean then, if you need to do that?
But you could define lipid molecules as well as being particles associated with these smaller spaces if you wanted to look at interactions between different lipid subtypes and proteins, for instance.
I suppose you could, yeah. I mean, these basically, you could, yeah. I mean, it's sort of open to how you interpret your simulation. So you have this membrane that just defines the geometry. Typically, we view this as the lipid membrane. So the lipids are kind of the membrane, and then we just add molecules inside. But I mean, there's nothing that prohibits you from actually adding lipid molecules to the surface.
It might be easier to define a space as having a certain lipid content than having characteristics based on that.
Again, I think it depends probably on the real question that you have. So maybe if you're just wanting to study some receptors, GPCRs, and binding to them, then you may just want to place them in a certain region at a certain density that you know from experiment, and then just use that as your simulation and just say the lipids are, the lipids basically are encapsulated in the diffusion coefficient of these receptors on the surface. Right? So basically, the diffusion coefficient is kind of sort of captures all these effects that these molecules encounter whilst they're diffusing in the membrane.
But this gets down to one of the limitations of MCell.
Mm-hmm.
It uses point molecules.
Mm-hmm.
And if you make things small enough, then the size of the molecule
Yeah.
has to be taken into account.
Right.
In some
You mean for surface
some ways
molecules?
You can fudge it in various ways,
Yes.
but you have to do that.
Right. Yeah, clearly. I mean, if at some point you reach that limit, yes.
So
Yeah.
that narrow spaces, for instance, if
Mhm.
molecules are moving in,
Right.
that's comparable with the real size of the molecule. Then
Right.
you have to do something.
Right. Right.
So we are working on versions of MCell where the particles do occupy
Yeah.
and can really crowd each other and get each other's way. But it's still experimental, and we're still trying to characterize the correctness of it and everything. But it is something that we're.
Yeah.
It's also considerably more computationally expensive to do this. But if that's what you need to do, then that's what you need to do. So the timing is as equilateral triangles as
Yeah.
Right, yeah.
So if it's a bad shape, it will stay bad shape.
Yeah.
Right, exactly. If you want to, if that's a concern, then what you need to do is remesh your mesh
Yeah.
to be more nice, right? But MCell doesn't. MCell's not a mesh generator. It's a mesh consumer.
Yeah, if you get your mesh from some other source, it's good to take a look and maybe think about would it make sense to remesh some of it. Of course, the less vertices and triangles you have that still represent your geometry, the faster the simulation. So and then sometimes if you have really narrow triangles, that's just numerically also more difficult to handle. So it's in general good if you get your geometry auto-generated from something and you realize, oh, I have all these really narrow and long triangles, to perhaps remesh this into a little bit more benign type mesh. So that's definitely a good idea.
There's some the mesh generation is a whole other can of worms, so to speak. And there's a lot of pretty good. Well, not a lot. There's several pretty good mesh generators out there, and some of them actually plug into Blender.
Mm-hmm.
So you can actually do this inside one environment.
Right. Okay, so that said, basically, just to explain the syntax, so the way to actually define the orientation of your molecule in the surface, either the top at the front or the bottom at the front, is this syntax that we've seen here in this example previously. So basically, when we add the surface molecule to this release site on the surface, you see there's this apostrophe here. And the apostrophe tells MCell to place the molecule like so. So this actually instructs MCell to place molecules with the top domain at the front of the surface. Alternatively, you could have put a comma here. So surf comma would have placed it like so. So at this step, basically, at the point of releasing your molecule, you tell MCell which way you want them to be in the surface. And if you really don't care, you can give it the semicolon, which basically means random orientation, I don't care. And MCell will place each of the 2,000 molecules in this case at a random orientation. So we'll have probably around 1,000 pointing up, or 1,000 in the top front configuration, about 1,000 in the bottom front configuration, plus minus some. And right, sort of, yeah. Does that make sense? So basically, this is how you if you want to model, let's just say you want to model transport across the membrane of some transporter or channel that you have, then you would think about, well, how do I want to place my channel in the membrane? And we'll come back to that in a second when we talk about reactions. But basically note that at this point you have two ways of placing them, or just ignoring it. And the final thing I wanted to mention before the break is somebody doing exercises upstairs. So last important part is for us to define reactions. And reactions are specified fairly simply. You basically have reactants, you have products, and you have a kinetic reaction rate. So this would be the volume 1 reactant becomes the volume 2 reactant with this rate. You can have reactions like this, where two volume molecules combine, and the null is sort of a special reaction, which basically means they disappear. So I'm not sure what that means biologically, but this is sort of just an example of what you can do. So this is kind of magic, I guess. And then let's briefly talk about this kind of a reaction. And again, you can see we have these commas and apostrophes again. So as soon as you have reactions that involve a surface or surface molecules, you have to think about the orientations again. And so let's talk about this reaction here. So let's see. So basically, in pictorial way, and it's actually quite good to draw pictures because it's a little bit difficult to sometimes grok that in your head. So let's look at the reactant side. So we have a surface molecule called surf. And do you remember we placed it previously in the membrane like this, top to the front. So this was when we actually did the release site. Before we replaced it top front. So this is how this molecule is currently located in the membrane. And then we have a volume 1 molecule that has a comma, so it's the opposite of surface. And what this basically means is that the volume molecule reacts with the bottom domain of this molecule. So opposite sides means volume 1 reacts with the bottom domain of this molecule. If they would be on the same side, the volume molecule would react with the top domain. So that's what this notation means. And on the product side, you can see now we still have these two molecules. Now they're on the same side. That means the volume product is released on this side of the molecule. Right? So basically, this specifies transport of volume 1 molecule across the membrane through this surf molecule. So this is one simple example how you could actually model transport of some species, volume 1, through the membrane, using this notation of using the fact that you can actually orient these molecules in the membrane as you wish, and then use this syntax to either specify that volume 1 should react with the bottom or the top domain. So you can pump out, you can pump in, there's actually a lot more flexibility, but that just shows you what you can do using this syntax.
We would have exactly the same syntax if the surface normal was the other way around. That wouldn't matter, right? Because
Right.
you would just place the molecule the other way.
Right, right.
So it's just parallel or anti-parallel
Right.
with respect to each other.
Right, exactly. So if we would have placed the surface molecule the other way around initially with a comma, it would have basically pumped the molecule into the volume. And this is actually not the whole story. You can actually add absolute orientation to that, so there's no ambiguity. So in this case, this reaction doesn't actually tell. This reaction could specify either this case or this case, right, depending on how these molecules are on the surface. So these are both possible scenarios. But you can make this unambiguous using an additional kind of syntax. And if you're interested, we can talk about this. I don't want to talk about it here because it becomes even a little bit more complex. But I think the message to take away here, you can more or less, using the syntax, describe very complex scenarios of transport or what have you, receptor binding to one side and release to another side or both sides, using this syntax. So it's quite rich in terms of what you can actually express with it. Yes?
Is the reason why it assumes that it's an orientation versus which side of the surface it is? Is that simply because you placed it on one side of the you placed it?
Right.
So you might ask that very question: Why are we going to such lengths here and have such flexibility rather than come up with something simpler? And the reason is biology is this complex. There's a specific case of enzymes called flipases that go around looking for proteins that are oriented a certain way and flip their orientation. And if that were to happen, say to our surface molecule, and if it were a transporter, if the surface molecule were a transporter and a flipase made it flip, and if the reaction between Vol1 and that surface molecule didn't care about the absolute orientation in the membrane, it would still keep transporting after it got flipped. Right? And so, yeah.
It was considered a membrane protein, so to speak, that changes orientation, while the other one is considered a soluble molecule that goes out to the other side.
That's basically.
Is that simply how you placed it at the beginning? That is the only difference?
Yeah, because this one was defined as a volume molecule. So this basically forces this scenario. But you could also define reactions with two surface molecules where you maybe have that, you have two molecules, they come together, but you require that somehow this domain of this molecule can only react with the domain on the other side for some reason. So they can really only associate in this specific orientation and not in this orientation. So the fact that we have this scenario is only because this was defined as a volume molecule and this defined as a surface molecule, but you can use that also to associate any kind of volume molecules, and you require it only works if they happen to be that orientation or that orientation. Yep.
So how things interact is based on how you define them in the concept. You know, it's not a separate thing. You define it. Based on how you define small ones, for example, determines how it's going to interact with work.
So basically what this does, it specifies that these reactions are possible. If they will actually happen is a different question, right? So basically these reactions will only happen if there exist surface molecules of this type in this orientation, and if there's a volume molecule in that space on which this surface molecule is located, right? So basically if you would have placed the volume molecule out here, this reaction wouldn't happen, because this basically specifies I want to pump volume molecule from the inside to the outside. If there is no volume molecule in the inside, this reaction never, it's defined as a possibility, but it never happens. So basically, what happens in your simulation is both defined by the reactions that you define and where molecules are. So you could envision that initially, so if you imagine maybe this is your cube, and initially there's no volume molecules in the cube, so these reactions that would pump this molecule out would never occur initially. But maybe there's some other process on the cube, like an uptake process, that slowly pumps in these volume molecules from the outside, and then at some point this reaction may start happening, because you then start accumulate volume molecules inside and they might pump out. So did that answer your question? So basically these reactions specify things that could happen, but if they do happen, depends on your model. So it depends on where are molecules located as a function of time. So it could be that this reaction doesn't happen for a long time until you accumulate volume 1 in this case in the inside, for example, or something else fires, or they get released from some intracellular store or something like that.
Would you like to comment at this point on the interaction radius?
The interaction radius for diffusing volume molecules?
I mean, how close do you have to be to the site of two molecules in order for reaction?
So in this case, the volume molecule has to hit the surface where this surface molecule is located. So there has to actually be a physical, so as it.
Oh, well, in the case of surface molecules, if we go back to the diagram of the very center.
Well, it hits.
And Mark,
right. So
explain more the mechanics of.
So more or less. So if this is your surface, you can ignore, or let's look at this triangle. So this would be a triangle on your surface that could contain a molecule or not. Let's say it contains there's a surface molecule on this triangle. And so the reaction will happen if the volume molecule hits anywhere on this triangle, right? So basically, we track, I hit this triangle, then we'll test, is the molecule on this triangle? Is the reaction defined with this molecule? And if yes, then these things will get triggered. So you're not.
On surfaces, they're not quite point particles.
Right.
They actually have a little surface area
Right.
that's equal to,
right.
that comes from the grid density.
Volume molecules, when they have to get within a certain distance of each other, it's the same issue, and now you can define them.
Right.
But of course, how you define it is the problem.
Right. So.
But the probabilities are basically computed based on the surface area. So they basically are scaled based on the surface area. So the actual computation of the probability is correct and takes into account the size of this triangle, right? So if the triangle is larger, the probabilities change, and when they shrink, they change.
And I think you're going to cover the mathematics. I'm curious how that works.
Right. And the same for the diffusing volume molecules. There we have sort of an artificial interaction radius that allows us to compute probabilities. But again, the radius itself doesn't really matter. I mean, the probabilities are scaled. It's just a numerical way of actually detecting collisions and computing probabilities for these reactions. Did that answer your question? Or? Yes.
So I have a follow-up question on these volume molecules. So let's say you have two different species of volume molecules, volume 1, volume 2.
Yeah.
Volume 1 molecule is very, very slow. Volume 2 is very fast. They interact for a certain time and they diffuse together. Could you redefine the diffusion coefficient for volume 2 to get the diffusion coefficient of volume 1 molecule for a certain time? And when they dissociate, the diffusion coefficient of volume 2 molecule would go back to the fast rate before.
I mean, what you could do is you could define a third molecule that would correspond to the complex, and so basically, right. And so basically, you have A and B combined to make a C. The molecule C would have whatever diffusion coefficient the complex would have, and at some point they would fall apart again to become A and B, for example. So that would be one way to model this association, and then the complex has a different set of properties, and then they dissociate again. Yes?
You said the reaction on surface molecules based on like hitting with the volume molecule anywhere on the triangles. But did you mean the triangle meshed in subblender or the?
No, these subdivided. So
Pericentric.
right.
Small pericentric.
So you don't see these triangles that we're talking about here. So they're basically each of your mesh triangles is subdivided into these small triangles at this density. So you don't really see those. So they're right.
But, because you pointed at
right. That was
mesh triangle we used before.
It's a good point. So that was pedagogically bad. So these are not these. You're right. Yeah. So this is maybe this one, not at the right density though. Was another question? All right, let's see. Yeah, go ahead. Back, yep. This one?
Yeah, generally, I think the system is inherent with respect to changes coordinate when we have a uniform system. But can we say that here we don't, our system is not uniform because of the chemical digits on this surface? That's why it matters whether we put this one on which side first.
Yeah, I mean, in general, these systems, you don't have uniform concentration across your system, so that you can have localized gradients of things, localized releases. So it's in fact often quite non-uniform. So you don't have equilibrium across your system. So actually there's transport, there's release. Does that answer your question?
Yeah, I mean, because that T and E are different as far as like chemically are different with top and bottom. So it matters where we start with when we put that volume on.
Right.
Yeah, that's fundamentally why
Yeah.
we are trying to account for this.
Yeah.
And then for the case where the front and back of the membrane itself aren't interchangeable, there's also a syntax
Yeah.
for that as well that you haven't seen yet.
Any other questions about this? So I realize that this is it's fairly complicated. It's often good if you have these types of surface reactions to draw pictures, to actually draw your system, to see what's going in, what's going out, where's stuff, and then it's often fairly straightforward then to write down the reactions. But it can take a little bit of thinking about to get actually the locations and the transport or whatever you have correct. And I didn't mention this in the beginning, but it's really key that especially once you, in the beginning when you start setting up your simulation, to look at your simulation all the time. It's really easy to mess up a spatial simulation. You think you have it all figured out, everything's going into the right compartment, only to realize when you look at it that instead of going in, they're going out, or nothing's happening at all. So it's really critical when you set up your simulation to use Cell Blender and look at it and make sure every molecule, everything happens in the right compartment, molecules appear in the right direction, and transport is the right direction. So it's surprisingly easy to get that wrong. So I mean, I really can't emphasize it enough to look at your simulation. Once you're done with setting it up, you don't have to look at it anymore. But until you have reached that point, it's really critical, because the spatial aspect really makes things quite more complex to get them right. Topology, inside, outside, and if you have a complicated geometry, so that can get tricky quite quickly. So I'm not going to talk about this a lot. I think we have opportunity to talk about this more this afternoon in a session. So the partitioning, just in 2 sentences, is a very useful thing to do. You should all do it. It's basically a numerical way of subdividing the simulation space, so your simulations become much more efficient. And especially if you have large models, this can save you a factor of 10 or even more in simulation time. So if you have bigger models, it's quite important to spend some time fiddling with that. And I'll talk more about this afternoon. So don't forget about this. By default, it's not going to be optimal, unfortunately.
For the giant model, what I'm going to show you is literally speeds up by factors of 1 million.
Yeah. So I just want to spend 2 more minutes, and then we'll have a break because I'm a little bit behind. There's 2 types of output that you can generate. Reaction data output is basically you can keep track of the numbers of molecules anywhere in your system. And we do this using these count statements. So a count statement counts a certain molecule, in this case volume molecule 2, in a certain area of your simulation. World is a keyword for meaning anywhere. So this basically counts volume 2 molecules anywhere in your simulation as a function of time. Often you want to be much more fine-grained than that. So you can actually define what we call sampling boxes. You can put boxes in your simulation space that are transparent to molecules, but that you can use to measure the number or concentration of molecules in whatever space you want. So you could have a sampling box close to your channel, one that's further away, and then just measure the number of molecules in these sampling boxes. And you can more or less customize this to measure any kind of quantity you want in your simulation, on the surface and in the volume. You can also count how many reactions happened. You can keep track of when and how often molecules hit certain surfaces and at what location. So there's basically a lot of flexibility there to track whatever variables are important for you to analyze your model. Number of calcium ions in this space, or number of bound calcium ions to your synaptic vesicles, whatever you're interested in. And then the second output is visualization output. There's a couple of output formats. I think the one that most of you want to use is the Cell Blender output format that Cell Blender can read and then just visualize your model. And again, here you can specify the kinds of things you want to make part of this visualization output. We have a keyword, all molecules. It just gives you everything. It's a good thing for the beginning. It's probably not what you want maybe if you have a huge model. So you can also use this to specify, I just want my A molecules or only my Bs. So if you have huge numbers of molecules, sometimes you don't want to see them all. You just want to know, certain types of molecules. And you can also specify when you want to capture that data all the time, all iterations, or just at the beginning and the end, or every thousandth time step, whatever you want. So this is the way you would use it for Cell Blender, and then you can actually look at it in Cell Blender and play it back and kind of see what happens. And you should do that a lot in the beginning when you set up your simulation. Often it's a good idea, unless you use this output, this viz output for other purposes, to turn that off once you do production runs, because it generates a lot of output. So if you do 10,000 iterations of your simulation, you might not want to do that 10,000 times because you're never going to look at it. So it's often a good idea to turn that off once you're doing production runs. I know that there are people who actually use this type of information to actually get at the XYZ coordinates of molecules as a function of time. And for those, of course, they need this type of information. We also have an ASCII version of this data, which is basically just XYZ coordinates of all the molecules as a function of time. So for people that want to track molecules, that's quite useful for doing that. It's not the most efficient, and if there's people who really need to dump a lot of molecule data frequently, we can talk and maybe see if there's a more efficient way for us to provide that data, because ASCII is easy to access, but it's not super efficient.
The Cell Blender format is very simple. It's a binary format. It's very, very simple and probably as compact, except for possibly compressing it. But I don't think it would compress well. I think it's about as compressed as it can be.
Yeah.
And it's about as small, compact, and efficient as it can possibly be. It's very simple. So if you wanted to know what that format was, you could document that or tell you exactly what it is.
Yeah. OK, I suggest we'll have a 15-minute break or something, and then we'll soldier on.